Today I would like to present the fundamental principles of treating cancer as per naturopathy, that is detox and oxygenation. This is actually a review of previously conducted various researches. Healthy human body don't entertain cancer, so it is altered biochemistry of cells which initiate, grow and spread the tumor. Bringing the body back to the healthy homeostasis is the key factor to prevent and to treat cancer. Detox and oxygenation not only treats cancer but also increases the efficiency of the cancer-specific targeted therapies of various alternative medicinal systems like Homeopathy, Ayurveda, Siddha and TCM.
Excuse me, Dr. Santhosh. Do you mind, do you have the slides for that?
Yeah.
Do you mind using the slides as well? Thank you.
Yes, exactly. First I go with detox. Cleansing the cellular microenvironment allows the natural healing mechanisms of the body to restore functional balance. The detox process helps to change the body's biochemistry so that it no longer becomes a suitable host for cancer cells to feed upon, grow and spread. I'm going to present 4 chief growing grounds of tumor, namely excess glucose, inflammation, sodium and mucus, which needs to be detoxed in order to successfully manage the cancer. First one is the glucose. As per Warburg effect, all cancers expressed high rates of fermentation, which is linked to mitochondrial dysfunction and genetic mutations within the cancer cell. These defects cause cancers to rely heavily on glucose for energy. Cancer cells need 19 times more glucose than non-cancerous cells. This graph shows high the cellular glucose level, less the chances of survival in a study with mice, which is an indication to detox excess glucose from the cells. Not only that, the tumor suppressor gene P53 through DNA damage and hypoxia leading to growth arrest and apoptosis. Inactivation of P53 found in 50% of human cancers. Glucose restriction induces P53 mutant deacetylation, rooting it for degradation via autophagy. Calls the importance of glucose detox. Here you can see the difference between the normal wild type P53 and mutant P53. Another carcinogenic activity associated with glucose is impaired PI3K pathway. Insulin action taking place mainly through PI3K pathway. Cancer cells show mainly hyperactivity of PI3K pathway and increased glucose uptake. A low carbohydrate diet, which is known to increase insulin sensitivity, can correct the central transmission of this pathway. So, glucose itself, P53 mutant expression and impaired PI3K pathway are undoubtedly points to a glucose restricted microcellular environment for the cancer cure. Second one to detox is inflammation. Many cancers arise from the site of infection, chronic irritation and inflammation. The tumor microenvironment, which is largely produced by inflammatory cells, is an indispensable participant in the neoplastic process, fostering proliferation, survival and migration. Hence, anti-inflammatory detox protocol is an inevitable step towards successful cancer management. Next one is excess of sodium in the cells. All chronic diseases, including cancers, starts with the loss of potassium from the cells and resultant invasion of sodium plus water, causing loss of electrical potential, improper enzyme formation and reduced cell oxidation. Hence, detox aims to excrete excess sodium may check the tumor growth. Next ones to be detoxified is mucus. Mucin 1 has been shown to affect tumor invasion and neovascularization, interactions with the vasculature and survival and growth. Due to the prevalence of mucin 1 expression in the metastatic disease, flushing out of excess mucus through proper detox could promise a great important improvement in the treatment of cancer. Now I have shown mainly what are the items to be detoxified in connection with cancer. Before to explain how to do it, I move to the importance of oxygenation in the treatment of cancer. Hypoxia, a common phenomenon in the majority of malignant tumors. Hypoxia leads to dysfunctional vascularization and acquisition of epithelial to mesenchymal transition phenotype resulting in cell mobility and metastasis. I request your attention towards this table. It's a study conducted by Barbara Moose from Washington University School of Medicine. Shows the difference between the oxygenation measurement of normal tissues versus cancer tissues. See, every cancer shows this hypoxia as a common characteristics. For example, brain 4.6 normally, but in tumor it's 1.7, and in lung 5.6 normally, and with the cancer it's 2.2. Hence, I strongly believe that hypoxia is the firstmost problem to be addressed in the management of cancer. Before starting with targeted therapies by alternative medicine practitioners, if the oxygenation of the cells can be increased, then the efficacy and the bioavailability of the target medicine and therapy would be wonderfully high. I would like to speak about two important hyperoxygenation therapies and antioxidants. First one is hyperbaric oxygen therapy. Hyperbaric oxygen therapy involves administration of 100% oxygen at elevated pressure. It increases plasma oxygen saturation, which facilitates oxygen delivery to the tissue. Hyperbaric oxygen therapy alone has been shown to inhibit tumor growth, reduce tumor blood vessel density, and reduce the preferential expression of anti-cancer genes. Next one is the administration of hydrogen peroxide. Potential therapeutic use of hydrogen peroxide can decrease the proliferation and increase the apoptosis. In addition, metastasis may be prevented because of the inhibitory effects of the hydrogen peroxide in cell migration and invasion. Artificial hydrogen peroxide production detected to cancer cells in an excessive manner may lead to cancer cells to death. Here I would like to show you a picture. This is a study conducted by Teiki Takoko from Nagoya City University Graduate School of Medical Sciences, Japan. The first A is the control where only sodium hyaluronate injection is given. The second one with sodium hyaluronate injection, hydrogen peroxide also has been given. So with this, the pulmonary cell positive areas were significantly lower than the control group injected with H2O2. So, but with the problem with hyperoxygenation therapies, it may generate a large amount of free radicals, which could increase the tumor growth. So, protective against the possibility of free radicals such is fairly simple with the use of antioxidants. A lot of supplements like superoxide dismutase, glutathione peroxide, methionine reductase, and catalase are strong antioxidants. Naturally, vitamin C, wheat and barley grass juice, then brassica genus vegetables recommended by National Cancer Institute of America for its antioxidant properties. So, regarding detox, to detox inflammation, the first one is the fasting. Fasting helps to reduce the inflammation by reducing the monocytes and oxidative stress. Next one is the lymphatic massage, which also helps to decrease the swelling from the body. And herbal preparations, curcumin, mistletoe, and boswellia frankincense helps to detox inflammation. Next one is to detox sodium. We have sauna as well as diet asparagus, corn, and potassium-rich diet, which helps to push out excess sodium from the body. To detox mucus, fasting also helps along with diet. Radish, apples, and oats helps to detox mucus from the body. The next one is the glucose is very important. Here I would like to give only a single remedy, that is ketogenic diet. Ketogenic diet is a low carbohydrate and high fat diet which decreases blood glucose and elevated blood ketones. Ketone bodies can serve as an alternative energy source for those cells with normal mitochondrial function but not for cancer cells. Many clinical data suggest decreased glucose delivery to the tumor as a causal mechanism in the ketone diet. And now I would like to conclude with an exciting, promising, and positive research conducted by Angela M. Puff, Department of Molecular Pharmacology, University of South Florida, about the ketogenic diet and hyperbaric oxygen therapy. In this research, 40 adult mice were taken. On day 0, 1 million tumor cells were inoculated. They were categorized in 4 groups. First one is fed with normal diet. Second group, normal diet with hyperbaric oxygen therapy. Third one, ketogenic diet. And fourth one is ketogenic diet with hyperbaric oxygen therapy. We can see here the result. The survival rate time of the control group is 31, whereas the last one is 55.5 days. And the tumor luminoscent study, the first one is the control group, the last one, almost the tumor is reversed by these two modalities. So I am positively convinced that with this combination, if we can incorporate intermittent fasting for complete detox and adding cancer-targeted homeo or Ayurveda medicine, we can successfully manage almost all types of cancers. I'm also actively looking for a new venture of a clinical practice based on integrative oncology where I can use my knowledge. With that note, I conclude. Thank you.